This video has been produced as part of the Classroom Learning Series for Green and Healthy Schools, Wisconsin.
Hello, and thanks for joining us today. My name is Nicole Filizzetti, and I'm here with the LEAF program at the Wisconsin Center for Environmental Education. In today's webinar, we're going to talk a bit about how to take a look at the diversity of a forest plot and how observing a forest in winter can help you learn more about the functions of that ecosystem. Before we get started, I'd like to take a minute to think about whose land I'm on as I do my work and live my life today. I encourage you to take a look at this map and do the same, and to take a moment to think about what this means to you. I acknowledge that where I live is the historic homeland of the Anishinaabe people and is currently the home of the Lac du Flambeau Band of Chippewa. I'm grateful for the care that they and other Indigenous people have given to this land and to these forests since time immemorial, and appreciate the ways their traditional knowledge affects my understanding of sustainable forest management and the work I do in education. This is the fourth and final installment of LEAF's Understanding Community Systems series. If you are interested in any of the other topics, be sure to contact us so we can direct you to the recordings. They're all found on the YouTube channel for the WCEE. To give you an idea of what to expect in our half hour together, here's a brief look at the topics we'll be covering. We will be focusing on 3 components of forest diversity and demonstrating some fun and easy activities that you can do to learn more about the forest in winter. For our field activity, we'll be following a procedure from LEAF's forest diversity lesson. In this activity, students study and collect data on 3 components of diversity found within a forest: species diversity, structural diversity, and functional diversity. We'll talk about each of these 3 components a bit more in depth throughout the webinar. As you're watching the video of the field experience, I encourage you to think about what my colleagues are doing that are examples of science practices. When you do this activity in your own backyard or school grounds, you'll be engaging in the real work of scientists. Without further ado, I think it's time to head out to the field with Kate and Steve.
A hike through a forest after a fresh snowfall provides a peek into how the ecosystem is functioning as a habitat. Animal tracks can tell you what critters live there, what direction they're headed, and what they might have been doing. It's exciting to do a bit of science detective work.
Here are a few resources you could use to help sleuth out the answers. Tracks and other signs of animal activity tell the story of the forest. Here are a few other examples. The species found can provide insight into how healthy the forest is. Similarly, the type of forest creates habitat suitable to specific critters, so scientists can then predict which species should be present.
Hi, my name's Kate Flick with the LEAF program, Wisconsin's K-12 Forestry Education Program.
And I'm Steve Schmidt, and I'm happy to be presenting with Kate this forest diversity activity.
We're really excited because we're here at the Central Wisconsin Environmental Station with the University of Wisconsin-Stevens Point College of Natural Resources. We have Sunset Lake over there, Minister Lake over there, and we're going to go visit 2 different ecosystems today.
Hi, we're here at our first sample plot where we're going to study forest diversity. And what's nice about this location is we have a red pine plantation behind us, and on the other side of the camera is a lake, and in between the two it transitions into a mixed hardwood. So there's a lot of change taking place in a relatively short amount of time. And we're standing on the edge of a plot that we just set up that is 33 feet by 33 feet, and which is 10 meters. And when you're studying woody tree species, in science, that's the size plot, sample plot that is pretty much a standard, and we're getting ready to collect our data.
And now back to the studio with Nicole.
Thanks, Kate. Now that Kate and Steve have their sample plot set up, they're going to be making observations and collecting data related to species diversity. Species diversity is measured by looking at richness and evenness. Richness is a measure of the number of tree species in an area, and evenness is a comparison of the number of trees of each species. You'll see that Kate is only going to be counting trees that are greater than 4 inches in diameter, and she's going to show you a cool trick to estimate what 4 inches of diameter looks like, so you don't even need a measuring tape. Let's talk for a minute about why species diversity matters. Diversity is the way that ecological systems exhibit resilience. When a disturbance occurs, such as a disease or a destructive wildfire, how the forest bounces back depends directly on species diversity. In general, biodiversity can be equated with the health of an ecosystem. In monoculture forests, or forests that are made up of only one type of tree, a disease that comes in has a better chance of killing more of the trees. For example, some urban forests in Wisconsin have mostly ash trees. The invasive emerald ash borer is destroying these forests, forcing towns to cut them all down and replant with younger trees that will not soon provide the same ecosystem services that the ash tree once did. Foresters are replanting with biodiversity and resilience in mind. Think about this as you see more about Kate and Steve's 2 forest plots.
All right, so the first parameter that we're going to take in is species diversity. And for species diversity, we're going to be looking at richness and evenness. Richness is just a measure of how rich is the plot. Are there a lot of species, a lot of different species, or is there only one species? Evenness is just a measure of how those individual species are distributed throughout. So is there 100 red pines and only 1 oak? That would be a very uneven stand. All right, so to take species diversity, we're just going to look at all the different species that are in our 10 meter by 10 meter plot. But we're only going to count the trees that are 4 inches or more in diameter. An easy trick for this one is to take an 8.5 by 11 piece of paper and fold it in half. So that's about 4 inches.
And just note, we're going to do this at diameter height in forestry, which is about 4.5 feet. You might want to think about where 4.5 feet is on your body, measure it out, and then use that as a reference point.
You can see that if this tree were in our plot, this one's a little bit less than 4 inches, so we wouldn't count it. This one, however, is greater than 4 inches, so we would count that one. And we're just going to look and see what are the different species in here. You don't necessarily need to know what the exact species is, so you don't need to do tree identification. You can just observe that they're different. So like, oh, this bark is different than that bark, or this leaf is different than that leaf. Though we do have a couple other tools that we'll talk about a little bit later if you do want to get into tree ID. Should we go explore our plot?
Let's go.
So here we've got one tree that is a red pine species. So that's one. This one is a different tree, but it's too small, so we're not going to count that. Here's another one, too small. Here's another one, red pine.
Do that for all trees in your plot greater than 4 inches in diameter. As you're collecting data, you might run into a couple things that you have questions about in the field. Here's a few. If it's a stump, don't count it. If it has multiple stems, that only counts as one tree. And if it's a dead tree, you don't count that one either. So let's take a look at the data. This is some data that's comparable to our 2 sites that we have. You'll notice that in the plantation, there's only one species. It's red pine. And while there's 13 red pine in our plot, that means that there's only one species, which makes it not a very rich plot, and it's very even because there's no other species in that plot. Now in our second plot, which you'll get more introduced to in a second here, you'll notice that there's 3 different kinds of species and a total of 7 trees. So in richness, we say that there's 3 species, which makes it a bit more rich than our plantation. And in terms of evenness, we have 2 white pine, 4 maple, and 1 hemlock. That means that it's a little bit uneven, But if I were putting this into a data category, I'd probably shift that uneven a little, like 2 or 3 hashes over, because it's not a totally uneven stand.
Now that we've considered the diversity at these two plots, think about how they'll be different in winter. How might the wildlife signs be different? What signs might be more apparent in winter than other seasons?
And now back to the studio with Ron Burgundy. Oh, I mean Nicole.
Now that Kate and Steve have investigated the species diversity of their plot, they're going to look at the next category, which is structural diversity. If you think about the word structure, you know that it refers to the way that something is built. So structural diversity is about how the forest is built, specifically in terms of layers, like overstory or canopy, and understory, which includes smaller trees as well as shrubs and ground cover plants called forbs. More layers means more structural diversity, and fewer layers means less structural diversity. How does this affect the habitat potential of a forest plot? And how does structural diversity change in the winter? I want you to think about these questions as you watch what Kate and Steve are doing with this second set of observations.
We're at our second location here, and we're going to compare the structural diversity, which is one part of the ecological diversity. We're going to compare the structural diversity from our first site, which was a pine plantation, to our second site here, which is a mixed hardwoods location. And when we're doing structural diversity, we're looking at the layers up through the forest. So we've got up above, we've got the canopy or the overstory, and under the canopy, we've got our understory. So we've got smaller trees that don't make it quite to the top. And we've got shrubs, which there were a lot more shrubs and raspberry bushes at our previous location. And then we've got the forbs layer near the ground, which here we've got some examples of ferns. In the spring, there might be some wildflowers and things like that. So basically herbaceous plants rather than woody plants. So when we're looking at the structural diversity, not only are we defining these different components, but we're also looking for signs of wildlife, because that difference in structural diversity creates different places for animal habitat. And so we've seen some interesting habitat out here besides your typical deer scat. We've also found signs that beavers have been eating on the aspen, and we've got some footage of that, and also some buck rubs from when the deer are moving through. We've also heard some nuthatches and chickadees. So what we're trying to do is note in each layer where those different signs are, and that helps us look at the structural diversity and compare that from one plot to the next. The structural diversity data are grouped by the forest layers. First note whether each layer is present, and then explore and record what is observed. Here's the sample pine plantation data from the leaf guide. In this case, there were only 2 observations. Notice there were more layers present and more observations in the mixed forest leaf guide data. One would expect more structural diversity to equate to more wildlife observations. Be sure to listen for vocal animals such as birds, and look closely for our smaller critter friends such as small mammals, insects, or other signs such as spider webs.
Hey Steve, before we go back to the studio, I really like imagining what that looks like in winter, from the forest floor to the shrubs to the understory and then all the way up to the canopy. What does that mean for wildlife and everything in between? And now back to the studio with Nicole.
The last component that we're going to look at is functional diversity. You know that a function is what something does, or the work that that thing is responsible for. So functional diversity is looking at the work that is happening in the forest. More work makes a more diverse forest. We measure this by looking in detail at the forest floor to see how much decomposition is happening in the duff, which is under the fresh leaf layer. If the duff layer is thick and decomposition and nutrient cycling are happening slowly, this means that forest plot has a relatively low measure of functional diversity. But if the decomposition is happening more quickly and that job is getting done more efficiently, that forest has a higher level of functional diversity. How does the rate of decomposition change in the winter? And how does the rate of decomposition in colder northern climates compare to places that are warm and moist for more months of the year? I want you to think about these questions as Kate and Steve are exploring their forest plots. And don't forget to be looking for examples of the practices of science.
We're going to talk about functional diversity a little bit and just experiment with one function that's in a forest. A forest has tons of functions. It's doing stuff with water, it's doing stuff with carbon, but we're going to look at nutrient cycles and the function of nutrient cycling. So different forests have different rates of nutrient decomposition. Decomposition is just when all the little critters and fungus and other things come around and they take the leaves from the fallen leaves from the trees and the sticks and other things that you can see around here, and then they break it down so that the plants can access those nutrients. If there wasn't any decomposers, the forest floor would just keep piling up until it was like above our heads and above the trees. But since we have those useful decomposers, we can see how one of the functions is nutrient cycling. This leaf has nutrients in it. Different leaves have different nutrients, and different other things like sticks and branches have stuff like lignin and other compounds that are kind of hard to decompose, so they take a little bit longer. So by looking into this little square here in the forest, we can get an idea about decomposition and nutrient cycling here in these different plots.
So stand at the corner of your plot, and while I like ultimate frisbee and disc golf, this is probably not something you should do. Instead, just stand there and have your square, toss it in there, kind of nice and gentle. If you don't have a square, you can use anything that is just marking a spot there. So for this, you want to measure at the top of the leaf litter down to the bottom soil layer. And the soil layer starts where there's bare mineral soil, so things like sand and things like that. So just kind of brush away the leaves, stick in your ruler, and see what the depths are. So we're just removing the leaf layer. You can see that it's a pretty thick leaf layer. There's the next layer of duff, which is only partially decomposed.
Whoa. Neat.
Look at that crazy thick duff layer. That kind of signals pretty slow decomposition. So just walking through that again, here we are digging down through the duff layer. We've got a whole bunch of pine needles, some other leaves, some sticks and twigs, and you really got to get down to that bare mineral soil. And right there where my thumb is where we're taking the measurement. So that's about 11 centimeters. And you can see that that's a big duff layer there. Slow decomposition, slow nutrient cycles. So here in our mixed hardwood, we're just going to brush away the leaves. You can see that we're hitting that soil layer a lot quicker, and there's not as much built up duff layer. That's signaling that the decomposition in this plot is happening at a faster rate, so stuff is more decomposed.
Really where we want to measure is if we're doing leaf layer, right?
Yeah.
So 3.
Yeah, 3.5. And then there's some pine needles, some oak leaves, and some maple leaves. And there's really not a duff layer, a well-established duff layer.
So when we look at the data between our two plots, this is probably one of the most distinct data entries. You'll notice that the decomposition in the red pine plantation is generally pretty uneven, and the level of decomposition is either low or medium, meaning that it's slow decomposition cycles, slow nutrient release cycles, and those layers are really building up in the forest. In our mixed hardwood, you can see that the depth of the leaf litter to the bare mineral soil is not as deep by a far cry. The composition is a little bit different, and it's a much more even decomposition, making that level of decomposition somewhere between medium and high. That means that the nutrient cycles are going a little bit faster in this mixed hardwood plot. So that's one way to measure and analyze different types of biodiversity in our forest systems. As we transition to a winter ecology landscape, just keep imagining all those diversity connections in our forest systems. And now back to the studio. Thanks, Nicole.
Well done, you two. At the beginning of the webinar, I encouraged you to think about the practices of science that were being demonstrated by the activity. Using the language of the next generation science standards, you can see that there were 3 important things Kate and Steve were doing. They were planning and carrying out investigations, analyzing and interpreting data, and obtaining, evaluating, and communicating information. So if you thought they were just playing in the woods, now you know better. Now that we've shown you how to collect data on 3 different measures of diversity, it's your turn to go out and explore. You can do this in your own backyard, a park in your neighborhood, or maybe even with your classmates at your school forest. It's fun and easy and doesn't really take any complicated tools. Here are a couple resources that might be useful for this activity. If you want some resources about tree identification, including a key that can help you identify trees in the winter by looking at their buds and branches, you can find those at our website, www.leafprogram.org. Teachers, don't forget about the trunks and kits that we have on loan through the WCEE. You can find them if you go to our website or by Googling WCEE UWSP kits. Also, the lesson that today's activity is from is available as a free PDF download, so contact us if you would like a copy of that. My final note to you is a reminder to get outside this winter. There are amazing things in your own backyard. Thanks again for joining us today, and take care.